Okay, greetings everyone. I'm Siphendulo Zaza. I'm an MSc student at Rhodes University. I'm currently doing my master's in applied mathematics under the supervision of Prof. Temkeng and Dr. Mori. So basically today I will be sharing a bit of my work that I'm currently working on, which is basically learning to detect anomalies in acoustic telemetry data. So aquatic acoustic telemetry data is basically information collected through underwater sensors to track and to study the movements of underwater fish species. So in our case, we're working with the dusty corp fish. So basically, the aim of this study is to integrate AI with biological data. So one would ask, why are we studying these telemetry data sets? What is the problem with telemetry data sets? And why is this a problem? So basically, we are studying these telemetry data sets in order for us to be able to understand aquatic animal behavior and their movement in their natural habitats. So by analyzing these data sets, we gain available information about their migration patterns and their habitat use. So the problem with telemetry data is the tagging of a lot of fish in a small space-restricted environment, like an estuary. So that results into millions of detections that makes our data very complex and difficult to work with. So the problem with tagging a lot of fish in a small space-restricted environment like an estuary is the creation of false detections or anomalies. So usually these anomalies arise from sensor errors or signal interferences or maybe unrealistic fish movements whereby a fish would appear to have moved long distances in a short period of time. So these are kind of anomalies that we are having in this kind of data. So now we know that if these anomalies are left undetected in the data, they can introduce errors, errors that may lead to misinterpretation of the data. They may also lead to incorrect conclusions and decisions. So now that's where the AI comes in now, whereby we want to try and automate this process of anomaly detection in acoustic telemetry data set. So basically we want to try and analyze this data in real time. So basically, this is the data collection process, whereby we had 50 dusty corbs that were surgically implanted a transmitter, and we also deployed 16 receivers in the breed estuary. So this is basically how this works. So we have a fish, and then we surgically insert a transmitter. And then so when it moves in the estuary and it sends signals, so when it passed or it's close to this detection range of this receiver, it gets recorded, the fish ID, the date, time, and other features. So this is basically our breed estuary in the Western Cape. So these are basically our receivers in the breed estuary. So these receivers are placed in a strategic way that a fish cannot pass multiple consecutive receivers without being detected. So from our domain expert in the ecological field, so they gave us these 3 criteria of anomalies in this data. So basically, the first one is that if an individual fish stays or is detected at only one receiver for the whole period of the study, we regard that as an anomaly. And you can see that this is for the first criteria whereby a fish is detected at only one station. The second anomaly criteria is that if an individual fish is moving up and down the estuary, as you would expect it to be, but suddenly stays at one location for a long period of time, we regard that also as an anomaly. And we can see that here, in this case, the fish was moving down the estuary from station 5 to station 1, and then it goes up to station 2, and then it stays there for a long period of time, and then we regard that also as an anomaly. And the third one is that if an individual fish is moving up and down the estuary, as you would expect it to be, but now suddenly it appears kilometers away in receivers that do not make any logical sense. For instance, here we can see that the fish is moving up and down the estuary from 5 up to 1, and then it's detected consecutively by these receivers. But now here we can see that it's from station 1 up to station 5, meaning it has skipped these consecutive stations. And I've mentioned that our receivers in the estuary are strategically placed in a way that a fish cannot pass multiple stations without being detected. So we regard this as an anomaly. So as I've mentioned, that telemetry dataset is a huge data with a lot of features that makes our data very complex. So these data sets require a lot of computational power when we are training this kind of data. So in our case, we had about 3 million instances where 91% of those data points were normal data points and 9% of them were anomalies. So if we had to apply our AI models into this kind of highly imbalanced data, then it will cause some problems. But luckily for us, we have what we call unsupervised machine learning models, which are capable of working with imbalanced data sets. So the purpose of an AI model in this case, in this context, is to train an AI model that will be able to accurately distinguish between normal behavior and anomalous behavior. So these are our machine learning models that we applied in this work. So basically, within the machine learning models, we have what we call shallow models or what we call simple models. So these shallow models are machine learning models that are adapted for small, I would say, non-complex data sets. But in our case, we're working with a huge data set with multiple features, which makes our data set very big and very complex. So these shallow models would generally fail because they are adapted for small, non-complex data sets. For the purpose of this study, we used this complex model, which is the neural network autoencoder, mainly because it can work with highly imbalanced data sets without any need of extensive data augmentation. So this is basically our autoencoder and how it works. So basically, first we just do the data preprocessing to remove any noise on the data, and then we put the data into an encoder as an input. So what the encoder does, it reduces the dimensions of the input into a compressed form, and then the compressed form now is fed into the decoder as an input. So the decoder, what it does, it tries to reconstruct the input. So now we have what we call the reconstruction error. So the reconstruction error is the difference between the input and the reconstructed input. So the aim of the autoencoder is to minimize this reconstruction error. So here we can see that we also need to have a threshold whereby we are going to say that, okay, if this reconstruction error is greater than the threshold, we regard that as an anomaly. And then if it's less, we say that this is a normal data point. And basically this is what I've just explained above, but this is just the math behind, which is we have the encoder and the decoder. So we can see that the encoder compresses the input into the compressed version, and then the output from the encoder now is fed in to the decoder to try and reconstruct the input. So this is how our reconstruction error is calculated, which is basically the difference between the input and the reconstructed input. So as I've mentioned above that, the autoencoder, we want to minimize the reconstruction error. So in order for us to be able to say that, okay, our model is actually doing well in terms of reconstructing the input, so the reconstruction error should be small. So now let's quickly go to the results because I don't have time. So if we look at this table, we have 3 rows, which is 1, 2, 3. So here we have resampling. So here on the first row, we have original data without any augmentation. On these 2 rows, we try to augment the data using 90-second resampling rate. and then we use 65-second sampling rate. So we sampled the data in order for the model to have more data to learn so that they can be able to do better predictions. So if we have a look at this first row, so let's start with the isolation forest. So this pink color represent normal data points that our model correctly predicted as normal. This color represent normal data points that are incorrectly predicted as anomalies. And then this one represent anomalous data points that are incorrectly predicted as normal. And then this one represents anomalies that are correctly predicted as anomalies by our data. So for all these confusion matrix, so this is the same. So basically, if we can look at the isolation forest, we can see that our model is doing a good job in terms of predicting anomalies because we don't have any misclassification of the normal data as anomalies. But now, if we come here, we can see that we only have 142 anomalies that are correctly predicted by our models as anomalies. And then we have this huge number that now is misclassified as normal, but actually it is anomalies. So this model, we can say that it is actually doing bad because the aim, we want to remove anomalies in the data, but now our model, which is this one, it misclassifies about 99% of the anomalies as the normal data. So for example, if we can relate this maybe into a medical field whereby you have a model that predicts if someone has a disease or not. So if your model is saying that someone who does not have a disease has a disease, so that particular individual can go for further investigation and then the doctors will actually see that, okay, this person actually does not have a disease. But now if your model is saying that someone who has a disease does not have a disease, then now they will not go for further investigation, and then now your model will be killing people. So also, which is similar to here, because we want to remove anomalies in our data, because we know that if we have anomalies in our data, it would lead to errors that will lead to misinterpretation of the data, and the misinterpretation of the data will also lead to misleading conclusions. So now if we go to the second model, which is the DB scan, we can see here that we have high number of correctly predicted normal instances, and then we have some portion of the normal instances that are incorrectly predicted as anomalies. And also here we have small number of correctly predicted anomalies, but we have huge number of misclassification of anomalies as normal instances. So basically, we can see that these, the DB scan and the local outlier factor, they are basically showing kind of the same thing because they are misclassifying a lot of anomalies as the normal data, which is not good. So if we go to the neural network autoencoder, we can see that we have a high number of correctly classified anomalies, normal instances as normal. So, but we also have some misclassification of the normal data as anomalies, which is 900. But now, if we go down here, we can see that now we have zero misclassification of the anomalies, meaning this model has correctly predicted all the anomalies in our data, which is basically what we want to do because we want to remove anomalies in the acoustic telemetry data. So now, let's go to these two, which is whereby we sampled our data, meaning we've added some samples to our data, which is we used 90 sampling rate and 65 sampling rate. So if we look at this first, which is the DB scan, we can see that DB scan and the local outlier factor, they show the same confusion matrices. So this suggests that these two models have reached their saturation point, whereby even if we add more data, it does not better our model. But if now we look at the isolation forest, we can see on the isolation forest that if we add more data now, we can see improvement because the correctly predicted anomalies are increasing from here, the original data and this augmented data using the 90 second sampling rate, and then here also we see some improvement. But this is a very minimal improvement. So basically, we still have a lot of misclassification of the normal data as anomalies. So this model also is doing bad. But now, if we go to our neural network autoencoder, we can see that we have some improvement whereby the misclassification of the normal instances is decreasing from 900 to 601, and then from here to 511. So now we can see from these models that the neural network autoencoder is actually performing better than these shallow models. And as we have said, that shallow models will generally fail in this big data that we are working with. So this is basically calculated using the above, so which will also tell us the same thing. So let me just go to the conclusion. So basically, integrating AI with biological data enhances our understanding of the marine ecosystems. So by automating the process of anomaly detection in acoustic telemetry data, now researchers can focus on analyzing the findings instead of doing manual data processing. And also these AI models are useful because they can be able to analyze large data sets in real time, which is something that humans cannot do. And furthermore, these AI models can be useful in predicting the future trends and providing providing early indications of any possible ecological disruptions. And now we go to the limitations of this work that we are currently doing. So the limitations of this work is because we are specifically working with the STICOP, and we are also focusing on the breed estuary. So if you would apply this model into a different estuary with different environmental conditions. So this model would need some recalibration or retraining to be able to maintain the high accuracy. So yeah, that's it. Thank you.
Okay, that's Mr. Sasa. Thank you very much. It's an interesting research. Do we have any questions for him? A quick one. Okay.
Yeah, just on the setting of thresholds for the models, what influences that, and what do they look like?
Yes. So so like basically the setting of the threshold, you are setting so for us we are actually maximizing the, okay, let me just, so for us, okay, let's see. So there are many ways to actually set the threshold. The other one will be based maybe from the from what is it? From the domain expert, and the other one will be to look for maybe the smallest reconstruction error here, because we are training the autoencoder on the normal instances. So if maybe we take the lowest reconstruction error and set it as a threshold here. So now if we have anomalies in our test data, so they will have more reconstruction error than this, then it will be greater than the threshold, then we will regard that as an anomalies. Yes. Okay.
Oh, is that a question?
Thank you. I heard you mentioned you working on getting a real-time data. So that means for me, you depending on memory cards and the batteries and all that. So my question is, how long, given the fact that we're still having a challenge of getting a network bypass the water waves and all that. So my question would be, how long does your batteries last and your kind of memory cards that you use and so on, if you are using that? Thanks.
So currently, we have not yet done that, but that's the aim of this work, because we are starting by creating a model, and then from there now we can be able to try and detect anomalies in real time. But for now, we have not yet implemented anything on that. So it's the future work that we are actually trying to work on in future. Yeah.
Okay. No, I think, yes, we have finally come to the end of the sessions. And just to thank every presenter, there is a lot of synergies. There is a lot of work that is being done. I think this platform gives us an opportunity to see the areas of collaboration. I have many questions I was jotting down there. I might not have asked, but I think I will personally have some contacts with the different presenters, just to have an understanding of some of the things. But there is a lot of collaboration that can come into this work, and I think we have a good opportunity within the AI, data science, machine learning environment, for us to be able to, we have students who are very creative, who can utilize this data to make informed decisions based on the data, but we are struggling to get the data sets that work for us. It's something which is quite key and which we really need to work on. Some people say the African problems should be solved by the Africans, but how do we solve them when we don't have the data? We know what is happening, but we don't have the data with us. So we need to put heads together and make sure that this data, it makes sense, is there. There is need for teams that can be established for us to have this data available for people to play around with and make informed decisions. Other than this, I think you have been a very good audience, and I was told to end at 4:30. It's 4:23. It's good time to close. I hand over to Kolan for any remarks.
No, just to thank everyone who attended this session. Believe you me, we should have finished this maybe 20, 30 minutes ago, but unfortunately the other gentleman went AWOL. So just thank you everyone for attending this one this time around. I'll see you maybe next year in June or July for the National Research Workshop in Pretoria. Thank you.
Give up.